The regulation of the acidity of a solution is very important to maintaining homeostasis. Acidity is based on the number of hydrogen ions in a solution. When an acid dissociates in water, it breaks into hydrogen ion and a negatively charged ion. As such, an acid is also known as a proton donor. Dissociation of a base in water produces a hydroxide ion and a positive ion. Hydroxide ions and hydrogen ions react to form water, removing hydrogen ions from a solution. This is why bases are called proton acceptors. Let's take a look at the pH scale. The pH scale expresses the concentration of hydrogen ions in a solution. The scale ranges from 0 to 14. A pH value of 7 is neutral. Any value lower than 7 is acidic, and any value greater than 7 is basic or alkaline. Let's explore various bodily fluids. Gastric juices are very acidic, about the pH of 1. Vaginal fluids about 3 to 4. Uterine and bladder much higher. Saliva is almost basic. And then you can see now we are getting into the alkaline environment for the rest of these. So this tells us that most bodily fluids have a pH of about neutral. Now let us take a look at the importance of strong and weak acids. A strong acid completely dissociates in water. There is a little attraction between hydrogen and chloride ions. Strong acids are not commonly found in the body. Gastric juice, however, contains hydrochloric acid, which is a strong acid. It is important for the digestion of proteins. Hydrochloric acid also kills bacteria ingested with food by destroying proteins required for its existence. Now let's take a look at the weak acids. Carbonic acid is a weak acid. It is formed when carbon dioxide is released into water. It is important in regulation of blood pH. A weak acid particularly dissociates in water because there is a strong attraction between hydrogen ion and a bicarbonate ion. Now let's turn to learn about buffers. Recall that too many hydrogen ions will break down the structure of a protein by breaking hydrogen bonds. The body therefore uses buffer systems to react with excess hydrogen ions. This protects the proteins from breaking down. Buffers consist of weak acids and anions from the salts of those weak acids. Let's learn more about that. This solution contains hydrogen ion and buffers a weak acid and anions from the salt of the weak acid. If hydrogen ions are added to the solution, the anion reaction or reacts with hydrogen ion. This returns the concentration of hydrogen ions to its original value. Likewise, if hydrogen ions are lost from the solution, the carbonic acid dissociates, releasing a hydrogen ion and restoring the hydrogen ion concentration. Buffer systems protect proteins by counteracting changes in physical environment.